Hello, are we live? Yes. Hello, everybody who's joined so far. We're just going to wait a couple of minutes to give everyone else a chance to join on. So I just realized I need to turn myself onto mute because I'm hearing my own voice back. And it's very distracting. How are you all doing? Where's everyone dialing in from? You suck at biology, so you're glad to be here. Well, this is the right place to be. We're going through proteins today. So I'm hoping by the end of this session, you're going to be an expert on that. A lot of people from London, Luton represent, Northwest. Nice. What's that like? Cumbria or Manchester? Dubai. You're winning so far for furthest away. Oh no, you've just been usurped. Someone from the Caribbean. That's cool. Which island? Ukraine. Pretty far away. Oh, Japan, a new winner. Pakistan, pretty impressive as well. Nigeria. Wow, are you guys making this up, or is this really this multicultural? I didn't expect that. And you guys you're doing A levels then? Are you in international schools, or your schools just do A levels? Space, Abdul, you win. Cool. Well, thanks for joining on a Sunday, giving up your Sunday afternoon to do some biology with me. Yeah, no, thank you. I guess you didn't have much choice with the new lockdown rules though, so that's probably why we've got more people. We're going to go through some stuff on proteins today, and we're going to talk you through the platform that we have, and hopefully you guys will be convinced to sign up. So without further ado, let's begin. So just a quick introduction. I'm Alex. I'm the head of biology here at SnapProvise. I've got a degree in biology, and I'm most of the way to my master's in conservation and wildlife biology, but I haven't quite finished it yet. And yeah, so I've been teaching for the last 4 years, and then have moved into tutoring now, and now also working at SnapProvise. It says Ollie on my video. Yes, Ollie was my predecessor, and yeah, we still haven't changed the name over yet. So it says Ollie. My name's Alex, but yeah, thanks for pointing that out. Cool. So today we've got some giveaways, some prizes for you guys. We're actually giving away a free account. AF2. Oh, thank you. We're giving away a free account for one person. So to be in with a chance of winning this, you just need to follow us on Instagram, and then you just say you attended this web class. Upload a picture and then tag SnapProvise, and one of you will be given a free account. So yeah, there's like 100 people in this thing at the moment, so you've got like a 1% chance of winning an account. So it's well worth doing. And for everybody, even if you don't win the competition, everyone's getting a coupon code at the end. So you're going to get a discount. What if you don't have Insta? I don't have Insta, so I'm not really sure what I'm saying about Instagram. So yeah, it's a bit prejudiced for people without Instagram, I guess. But you can just make an account and not put any photos on it, and you can still just tag it if you wanted to enter, and you don't have an account. Can I repeat which bit about the coupon code? That all will be revealed at the end. So we'll go through that after we've gone through the actual content. So we're going to start. Some of you might already be on the platform, so you might recognize this. The way we normally start a lesson, at least in biology, is we have this learning pyramid. So it's usually topics we've done recently in the last few weeks, kind of just like a reminder of what we're looking at to get us warmed up, ready for the topic today. The lesson today is 1 hour, give or take. We'll see how long. I'm usually really bad at finishing on time, and they're supposed to always be hour lessons, and I usually like run over a little bit. So I'll do my best to power through today. So first question is, and this is open to everyone, by the way. So I want everybody answering all the questions. When I throw out a question, it's not targeted to a particular person. It's for everyone to answer. So the more you get involved, the more you'll get out of the session. So like, yeah, please do answer if you know the answer. Obviously, if you don't know the answer, that's also no problem. So first question is, which molecule is required for a hydrolysis reaction? What are we thinking? This is for all exam boards. So AQA, OCR, both OCRs and EdXLs. Yes, wow, all the answers are flying in. Love it. Yes, water. I thought I'd start with a nice, easy one to get everyone warmed up. Perfect. So hydrolysis and condensation reactions, we're actually going to be talking about them a bit today. They come up time and time again throughout the biological molecules topic. So you need to have them super locked down. Next question is, what is an ester bond composed of? So slightly harder. I'm sure you guys, well, where do you, if you don't know that, can you tell me where would you find an ester bond? Maria, that's close, but not quite. Yeah, so you find ester bonds in lipids. So if you had a triglyceride, which looks like this would be glycerol. And this would be a fatty acid. And the ester bond would be between those. So yeah, ester bonds in lipids. Anybody know what the actual bonding looks like though? What would join the glycerol molecule and the fatty acid? If you remember, glycerol is just 3 carbons with 3 OH groups. It looks like that. And then a fatty acid has got a carboxyl group on the end. Yes, nice. I'm seeing the answers pop up now. So yeah, we've got a C-O-C bond, and one of those C's has a double bond to an oxygen on it. Cool. So next one, what is a glycosidic bond composed of? And if you don't know the answer, you can tell me where would you find a glycosidic bond? What sort of molecule would you find one in? Yeah, so we find these in carbohydrates. Yeah, sugars, carbohydrates. So you can be careful with saying sugars at A level. We kind of want to be saying carbohydrates more than sugars. Sugars is okay for GCSE, but better to say carbohydrate at A level. I'm also guilty of saying sugars sometimes, and it's easy to do. But yeah, better to use the proper terminology. And yeah, this bond is just C-O-C. So in both cases, the oxygen molecule is sort of acting as a bridge between the two molecules we're joining together. Yeah, it's different in nucleotides. Your dad, it keeps like making me double take every time I see it pop up. Your name. So where in the body would you find protein? And there's lots of examples. So yeah, what can you guys tell me? Liver, yeah. So yeah, muscle. Rather than saying liver, let's just say like all organs. What about specific types? Enzyme, yeah, nice. So enzymes are the most abundant in terms of diversity of all the proteins that we have in the body. So the vast majority of proteins that we synthesize in our bodies are enzymes. Because enzymes are so specific, you need a certain enzyme for a certain reaction. We need lots and lots of different types. Yeah, lots of great examples. Collagen, hair. Yeah, exactly. Keratin is the protein in hair. Channel proteins, elastin, insulin, yes. Hemoglobin, yeah, great answers. Hormones, yeah, some hormones are protein-based. Some of them are also based on, the steroid hormones are based on cholesterol. So not all of them are protein-based, but yeah, a lot of them are. Nice. So yeah, lots of great examples there. Next question, how are starch and glycogen different? Yes, who did dislike this video? You may leave. Yeah, glycogen is more highly branched. Good. Branched or more branched. Yeah. Starch is in plants. Good. Whereas glycogen is in animals. Plants, animals. And what they do in plants and animals is they're acting as an energy store. So it's sort of like your short to medium term energy store. Longer term energy store would be things like fats, which we store subcutaneously, so under the skin. Yes, nice. So starch is made up of two things, amylopectin and amylose, very good, whereas glycogen is just glycogen. Yeah, glycogen is more compact. Nice, Abdullah, exactly. So it's more compact because it's got more branching. So why does glycogen actually have more branching than starch? It's one of the reasons is to make it more compact, which is useful for animals because we've got to move and carry around all this energy with us. So for animals, having compact energy storage is more useful than for plants who just store it in their roots. They don't really go anywhere. So it doesn't matter so much that it's not compact. So we need it compact to move. And yeah, we also need fast energy release. So the metabolic demands of animals are way, way, way higher than plants. So having a very branched structure means there's lots of points for enzymes to attach to and to break down the molecule. So yeah, exactly. The branching is to do with 1,6 glycosidic bonding. So we should probably put that on there as well. But I don't want to go too much into this today because this is obviously carbohydrates and we're here today to learn about proteins, but it's all related, so it's all good. It's all good practice. Cool. So final thing is, can you tell me any facts about proteins? What do you guys already know? Proteins are cool. Yes. The different structures, yeah. Primary, secondary, tertiary, quaternary, good. Made of amino acids, nice. 20 different types, perfect. 4 levels of structure. You guys can teach me the class. Alpha helix, beta pleated sheets, very nice. Peptide bonds, perfect. They don't make DNA, but they are related to DNA. DNA is used to make proteins. So the process of protein synthesis, that's actually why we have DNA, to make proteins. Yeah, loads of great facts. Let's get started then. So we've got 3 objectives. Well, we normally have 3 objectives in a lesson. I think today we will probably only have time to get through the first 2, but we're going to look at the structure of amino acids. We're going to look at the formation of polypeptides by condensation reactions. And then if we've got time, we'll look at secondary, tertiary structure and maybe fibrous and globular, but I think we won't have time. This lesson ends at 6, give or take. And yes, I think it will be available on YouTube afterwards. So what we do in a normal lesson is we'd go through the spec points for everybody, but today, just to speed it up, I'm just going to skip over these. But if you want to screenshot your spec point as it comes up, please feel free to do so. That's the OCR one. That's Edexcel A and B. Edexcel somehow always managed to condense their spec points really nicely, so we can normally fit 2 on. Yes, I meant 7 o'clock, of course. Yes. So part 1, we're looking at amino acids. So hopefully, because you guys have already started the year, and this is one of the early topics, this should be familiar to most of you. So this is an amino acid, and there's 4 main parts we need to know about it. Well, I guess 5 if we include the central carbon. So we'll start there in the middle. So an amino acid, they're all based around this central carbon molecule. And then we've got 4 groups coming out from there. So we've got the carboxyl group, which you might recognize as the same group that's on the end of a fatty acid. So it's just a COOH if it's drawn out in the molecular formula. Yeah, have we got any A2 people? Sorry, any A2s? Or is it, we've got a mixture of year 13 and year 12? Yeah. Okay, nice. Also, the A2s, this might have been quite a while since you've seen this, so hopefully it's a good bit of revision for you guys. We've also got an amino group, so that's NH2. We've got hydrogen, that's just a hydrogen atom stuck on. And then the most interesting one is this R group. So we also call this R group the variable group. So why do we call this the variable group? Or what is the R group? Can you guys tell me? Yeah, it can change. So the R group differs between amino acids. And like you said, there are 20 different types of amino acids. And from these 20 different types, we can build infinite proteins, which is kind of cool. They're kind of like, think of them as like the building blocks of Lego. There's not that many different types of Lego blocks, but with Lego blocks, the things that we could make is infinite. You could make a different Lego model every day, and you still would be doing new things. So it's kind of like that. So from these 20, we build all the proteins that we need in our bodies and all the proteins that any organism needs to survive. So just, I kind of wrote this over here, but the amino group is NH2 and the carboxyl group is COOH. Do you have to memorize them all? Good question. No, you don't. You actually don't need to be able to name any of the amino acids, although, no, that's not true. OCR, you do have to know glycine and cysteine, and we'll go through them a little bit later on. Glycine is of note. Actually, yeah, we'll just do it now while we're on the page. Glycine is of note because it's the simplest amino acid. So the R group on a glycine is just hydrogen, and cysteine is of note because it's got sulfur in it, and that makes the disulfide bridges, which are the really strong bonds that hold together the tertiary structure. But yeah, other exam boards, you're lucky. You don't need to know any of them. Okay, let's look at some condensation reactions then. So to form dipeptides, which is 2 amino acids, or polypeptides, dipeptide, polypeptide, which is amino acids stuck together. These circles are meant to, well, they're not circles, hexagons are meant to represent amino acids, which maybe wasn't the best choice of shape because we normally represent carbohydrates with the hexagon structure, but just, yeah, let's go along with it, that they're amino acids. To form a dipeptide from 2 amino acids, we need to do a condensation reaction. So that means we're making bonds through the removal of water. So when we form our dipeptide, we're going to get H2O produced. And if we go from a dipeptide up to, we don't need to know the word like tripeptide or tetrapeptide, we just go straight to polypeptide. So 2 or more is a polypeptide. Again, we're producing a water molecule. Each time we make the condensation reaction and stick on the amino acid, we subtract a water. So let's actually draw out what that would look like. So we need to draw 2 amino acids. While I'm doing this, question for you guys, what is another word for polypeptide? Because we can use another word sort of synonymously with that. A polypeptide is a multiple amino acids together, yeah. What about a more snappy, yeah, protein? You can just, yeah, it's an example of a polymer. That's very true. But yeah, I was looking more for protein. So you can kind of use these words interchangeably. So we've got another amino acid down here. So I hope you guys can all draw out a amino acid. If you can't, you need to just sit down and learn it. It will only take a few attempts. Just like draw it out, cover it up, try and draw it out again. If it's wrong, repeat. And once you can do it a couple of times, it should be pretty much committed to memory. So to join these two amino acids together, we remove water. So the OH comes from the carboxyl group and the H comes from the hydrogen group. That gets taken away. And the carbon bonds directly to the nitrogen, like that. Just catching up with some comments. Don't you need hundreds to be a protein? Yes, technically, you wouldn't really get a protein with only like 3 or 4 proteins in real life. Would be many, many hundreds to even some of the big ones, like many thousands in amino acids in length. So because it was kind of difficult to represent on this screen, we've just done a simple version. Jeremy, yeah, I guess not exactly. Polypeptide and protein aren't exactly the same. A polypeptide chain is specifically the amino acids in a chain, and a protein is it can have a tertiary structure or a quaternary structure, so it's more like the folded up shape. But in terms of exam, when they ask this question in exams, when they ask like what is the primary sequence, you can say either answer for that. Cool. So next up, we're going to look at hydrolysis reactions. So this is the opposite. So the reverse of what happened last time. We're putting in water and we are splitting the molecule apart. So yeah, the definition for hydrolysis is the breaking of bonds through the addition of water. So I think just in the interest of time, I'm not going to draw it out, but if you can just picture the reverse process. So we've got this carbon bonded to the nitrogen here. So this carbon bonded directly to this nitrogen. Unlike in the ester bonds and the glycosidic bonds, there's no oxygen bridge holding them together. The bond goes directly from the carbon to the nitrogen. So it's a little bit different in a peptide bond. Should probably actually label that as well. Peptide bond. Yeah. So in the hydrolysis, it's the reverse. We add the water back in, we add the OH back to the carboxyl group, we add the hydrogen back to nitrogen, and they go back to separate amino acids. Yeah, exactly. Bond is peptide. These YouTube sessions, I've got a calendar at the end I'll go through. They're weekly in different subjects. So each subject comes up about every month, basically. Okay, so how are you guys feeling with what we've covered so far? Yes, there will be A2 ones. So I think the next one is going to be an AS one, and then the one after that for biology is A2. Good, lots of ones there, a few twos. Hopefully we can push you guys up to ones by the end of the session. I'm loving the emojis. Yeah, feel free to just respond with the emoji rather than the number. Perfect. So let's test what we've just looked at with some exam questions. Again, this is the format we would normally do for a lesson. We introduce a topic, we talk about it, go through some concepts, and then we test your understanding straight away with some exam questions. So it's really good to test that you have understood what we've covered. So first question, well, let's introduce the whole thing. So we're looking at an amino acid. So figure 1.1 is, hopefully you should recognize this as an amino acid. State the chemical symbol of the atom represented by the letter X. And most of you, yeah, exactly. Be careful, the people who've written out nitrogen, if it says the chemical symbol, you've just got to put N, and it should be a capital N. So some of you probably on the computer, you're not really thinking about lowercase or uppercase, but yeah, just be really careful. Don't write out nitrogen because it's asked for the chemical symbol, and make sure you write it with a capital because that's the proper chemistry way to do it. So next question, can you name the polymer formed from a chain of amino acids? Yeah, nice. Polypeptide. Yeah, and it is in the mark scheme to say protein if you like, but yeah. For this answer, I'd say probably stronger to lead with polypeptide. Nice. Next question. Name the bond formed when two amino acids are joined together. So, yeah, they're relatively easy questions. We've got some slightly harder ones coming up later. Peptide bond and description of formation. What are we going to say for that? Bit more of an open-ended question. Yeah, water molecule is lost to form the peptide bond, and more specifically, we get the OH from the carboxyl group, and we get the H from the amino group. Perfect. And yeah, exactly. Condensation reaction. Would be a good answer as well. Okay, next part, we're going to go through the different structures of proteins. So this is a really important part of proteins. We've got 4 different structures, and you need to know the bonding in each type and sort of how to define what they are. So the first one, we go from primary up to quaternary. So yeah, starting with the primary structure. The primary structure is literally just the order of the amino acids in the polypeptide chain. So in this colorful diagram, we've got lots of different circles. Each different circle is representing an amino acid, and each different color is supposed to be representing one of the 20 different types. One of 20 types. And yeah, the instructions for assembling these proteins in the order that they're in, this comes from DNA. So you transcribe the strand of messenger RNA in the nucleus that leaves the nucleus, goes to a ribosome for transcription. And we're not going to go into the details of it because, again, that's another class. But on the ribosome, in translation, the messenger RNA is converted into the polypeptide chain. So yeah, for our sort of definition, we would just say, oh no, sorry, the definition is at the top. So the definition of primary structure is the order that the sequence of the amino acids in the polypeptide chain, and the notes are kind of just 20 different types. Different types, and they can be assembled in any order. In any order. So you can have like one amino acid might repeat a lot of times in a row, or it could be completely random. But all of that comes from the DNA. Okay, moving on to secondary structure. Actually, one more thing to say here, because the bonding is really important for each of these structures. The only bonding involved with primary structure is peptide bonding. So these are the peptide bonds, and primary structure is held together by peptide bonding only. So these are examples of covalent bonds. Next up, we've got the secondary structure. So the secondary structure is held together by hydrogen bonding. Can anyone tell me what hydrogen bonding is, or sort of like how it works? Nadia, the covalent bonds were the peptide bonds. So peptide bonds are examples of covalent bonds. So a covalent bond is a strong bond, and it's formed by the sharing of electrons. You don't actually need to know the definition of a covalent bond for biology, but if you're doing chemistry, you'll be very familiar with it. But yeah, all you need to know for biology is that covalent bonds are strong, whereas hydrogen bonds are weak bonds. They're not very strong bonds. Yes, it's all to do with electronegative and electropositive. So we say that oxygen is delta negative. This means it's slightly negative. It's not fully negative. It's slightly negative. And hydrogen is delta positive. Again, it's not a full charge. It's not an ion, but it's slightly positive. And positives and negatives attract. And this sort of attraction between them is a hydrogen bond. So nowhere near as strong as a covalent bond. These are very easily broken. So when you heat a protein or you put it in a different pH, these are the bonds that break first. They're really, really weak bonds. So the two shapes we get, we have alpha helices, which are spirals. So yeah, the protein chain sort of like wraps up into a spiral, and we have beta pleated sheets. So much like a pleated skirt or a pleated curtain, it's like folded up, or imagine you folded up a piece of paper and then opened it up. The protein sort of, the polypeptide chain folds back round on itself, and it has this zigzag pattern. So those two structures make up the secondary structure, and it's held together by hydrogen bonding. If you can remember those two names and remember that's hydrogen bonding, that's all you need to know for secondary structure. Yeah, sorry, just catching up on the comments. Can it be both? Yes, in a protein, you will nearly always get both of these types of bonding. Sorry, both of these structures. So you'll get a mixture of alpha helices and beta pleated sheets. Yeah, good question. Zainab, what about the disulfide bridges? They are in the tertiary structure, which we're about to get to, like that. Ikra, no, you don't need to be able to draw it. You just need to be able to name them. Or it might be helpful to be able to recognize them in an exam if they sort of represented them with a diagram. Just think the alpha helices are helices and beta pleated sheets are this sort of folded shape like that. Cool. Any other questions? Or are you guys happy to move on to tertiary structure? Yeah, no worries, Ikra. And again, if we're in a normal session, there's quite a lot of you guys watching this one, so it's a little bit hard to answer all your questions. In a normal session, it's much smaller classes, so I can sort of answer all questions in one of the sessions on our website. It's a little bit tricky now. I'm kind of having to skip a few of them, but doing as many as possible. So the tertiary structure. This is like the 3D shape of a protein. So the alpha helices and the beta pleated sheets fold up to form the tertiary structure. Alpha helices plus beta sheets fold up to form 3D structure. So the three-dimensional structure is really important. It basically gives the protein its particular properties. So different proteins have got different things they need to do. So there's the big distinction is globular or fibrous. So whether they're structural or whether that's fibrous proteins, or if they're globular, they're things like hormones, enzymes. They're soluble. They go in the blood or in the cells. So the structure is really important, particularly for the globular proteins, which the major group of those is enzymes. As you guys will be aware, because I'm sure you've all studied enzymes by now, the shape of enzymes is key to their function. So the active site needs to exactly match the substrate that it's catalyzing. So yeah, this is really important. There's four types of bonding involved with the tertiary structure. We've got hydrogen bonds, we've got ionic interactions, Actually, I just call it ionic bonding. Disulfide bridges or disulfide bonding. You can use either word. Bonding. And we've got hydrophobic, hydrophilic interactions. And that's mainly just in globular proteins, this last one. And it actually helps hold their shape together. What causes hydrophobic, hydrophilic? We're going to go through each of these bondings a little bit later on. So yeah, we'll go through and explain exactly how each one works. But yeah, good question. So before we get into the actual different bondings, we'll just talk about a quaternary structure. So a quaternary structure is held together by the same type of bonding as tertiary. As tertiary structure. But it basically means that the protein has got more than one polypeptide chain. So can anyone give me an example of a protein that's got a quaternary structure? Yes, Lillian, unfortunately, you do need to know each bond and like a little bit about each one as well. Hemoglobin, good. That's everyone's favorite go-to one. I saw insulin in there as well. Nice. Antibodies, yeah, very good example. Glycine is just an amino acid rather than a quaternary protein. Keratin is a good one. Nice. Chitin, yes. Yeah, good. Collagen, nice. Hormones, again, some hormones are protein-based, some of them are lipid-based, so be a bit careful with that one. But yeah, definitely some hormones are. Nice. So yeah, more than one. So most proteins actually have a quaternary structure. Yeah, the majority. Some of them do just have a tertiary structure, but most of them are made up of more than one polypeptide chain. We sometimes have non-protein groups associated with proteins. Does anyone remember what those are called? Non-protein. Yeah, prosthetic groups. Very good. And can anyone give me an example of a prosthetic group? Nice. Iron. Yes. Where? Iron in hemoglobin. Yes, good. So hemoglobin has an iron ion. Bit of a tongue twister. So in a hemoglobin molecule, hemoglobin is made up of 4 polypeptide chains, and each one of those has an iron ion in the middle. So Fe2+, Fe2+, and so on. Each one of these iron ions can bind one oxygen molecule, so one O2. So each hemoglobin molecule can take a total of 4 O2s or 8 oxygens per hemoglobin. Cool. What exam board is this? This is all exam boards. OCR, A and B, Edexcel A and B, and AQA. You all need to know this. To be honest, most of the biological molecules topic, it's quite nice. It doesn't really vary that much between the specs because it's such a fundamental sort of core you need for understanding biology. It's the same. It's the same for all of them. There are some like slight differences, but in general, they're very similar. So we're going to talk about the different bondings in a little bit more detail. We've kind of already talked about a hydrogen bond. So we said it's delta positive and delta negative attractions. We said hydrogen and oxygen was one example. In proteins, we also get it between hydrogen and nitrogen. So nitrogen is also slightly delta negative. So we can get bonds forming here. So we've got amino groups will be attracted to OH groups, and they'll be able to form hydrogen bonds. The definition of prosthetic group is a non-protein thing. A non-protein part of a protein molecule. So something like an ion is a common one, a metal ion, which is attached to the protein, and it gives it some of its properties. The pink lines they're sort of representing the polypeptide chain. So these are, but it doesn't have to be hydrogen bonding. This is sort of like the definition of hydrogen bonding in general. So it doesn't have to be just in a polypeptide, but this is a polypeptide chain. We've got some OH groups sticking out, and we've got some amino groups. And these, these are attracted to each other. Exactly, slight difference in polarity. Nice. So next up, we've got ionic bonding. So these are interactions between oppositely charged R groups. Between oops, skipped on a bit. So these are full-on charges. So one group may be positively charged, another one may be negatively charged, and those will be attracted together. And that's kind of all there is to it. So yeah, these are stronger than hydrogen bonds. So they would be later to break. If you're changing the conditions of where a protein's in, so for example, heating it or putting it into a different pH outside its optimum pH, these would be later to break than the hydrogen bonds. Disulfide bonds would be the very last ones to break of all the bonds that hold together the tertiary structure. So these are covalent bonds. And what a covalent bond is, this isn't, you don't need to know this for A-level biology, luckily. It's just if you're doing chemistry, you'll be familiar with this, but it's the sharing of electrons. So the sulfur atoms, they want to have 8 electrons in their outer shell. They both have 6 electrons. So the green crosses here, they're representing this sulfur and its 6 electrons, and the pink dots are representing this sulfur and its 6 electrons. So they share these electrons in the middle so that both end up with a complete shell with 8 electrons. So they're both happy, they've both got 8, sharing is caring, and they're much more stable like that. So they form this, this structure. So this is a cysteine molecule. This is another cysteine. And yeah, normally it's sulfur bonded to a hydrogen. That hydrogen disappears and when it's in aqueous solution, and then the sulfurs join directly together. So that's the actual disulfide bond or disulfide bridge. Cool. So what else do we say on that? That's kind of it. Yeah, they form between cysteine, and they'll be the last to break. So if you're heating a protein or changing the pH, they would be the very last to break. They're it tends to be sorry, I'm just catching up with the questions. It tends to be irreversible because the folding of the protein is like a difficult process, and it needs to fold up in a certain way, otherwise it won't reform that. So if some bonds are broken and some aren't, it won't be necessarily be able to reform the exact same molecule. So they tend to be irreversible when you heat a protein. And again, if you change the pH too much, this also tends to be irreversible. So the last one, hydrophilic and hydrophobic interactions. What this means is some of the R groups are hydrophobic and some of them are hydrophilic. So what do we mean by that? What do these terms mean? Kayana, if that's how you pronounce it. I'm seeing it's like Diana with a K. Yes, you can also call them sulfur-sulfur bonds. Yes, hydrophilic, water-loving or interacts with water. Put that in quotes because it's, yeah, interacts with water, water hating. It's kind of like the direct translation rather than the properties. So yeah, hydrophilic, they interact well with water. They're able to form hydrogen bonds normally, or that's how they interact well with water. Hydrophobic don't form hydrogen bonds, don't interact well with water. So they try to sort of orientate themselves away from the water. So what happens in a globular protein, which are generally water soluble, you've got a lot of hydrophilic groups on the outside. So the blue dots are hydrophilic, the red dots are hydrophobic. The hydrophilic groups will hang out on the outside, interacting with water, making the molecule soluble. The hydrophobic groups will combine in their hatred of water and orientate themselves into the middle of the protein, away from the aqueous environment and the water. And that helps to actually hold the protein together. The hydrophilic ones like being on the outside near the water. The hydrophobic like being in the middle, away from the water. And it's a stable molecule from that. Philic and phobic just means love, loving and hating. From, I believe it's Greek, but correct me if I'm wrong. But yeah, the same words, they crop up in other scenarios. Like arachnophobic is like spider hating, you're scared of spiders. I'm sure you can all think of something ending with philic. So yeah, what decides if it's hydrophilic or hydrophobic, Jeremy? Is that what you mean? It's to do with being able to form hydrogen bonds. So if it can form hydrogen bonds, it is generally hydrophilic. So things like OH groups, lots of OH groups would make a molecule hydrophilic because these can form hydrogen bonds with water, so it can interact with water, it can dissolve. If it doesn't have any OH groups, so for example, like lipids, they're just long, lipids are long chains of carbons and hydrogens. Well, this is like a fatty acid. It does have a carboxyl group on one end, which it's got one OH group, but the rest of the molecule, and I've only drawn a very minuscule fatty acid here, it should be like way, way longer. It's got one OH group and it's got several tens or several hundreds of just carbons and hydrogens. So yeah, they don't dissolve in water. Cool. Yes, of course, phospholipids are a little bit different. So phospholipids, they've got two fatty acids and they've got a phosphate group rather than a third fatty acid. It's all bonded to glycerol, same as a triglyceride. The phosphate group has a charge. And this means that phospholipids are a little bit special. One end is hydrophilic, one end is hydrophobic. So we call that amphipathic when it's got both hydrophilic and hydrophobic properties. So they're a little bit special in the lipid world because they're partially hydrophilic. Okay, how are you guys feeling with that, structures and bonding? Give me an emoji or a number. Let me see what you guys think. Is an ionic bond hydrophilic? No, it's a different type of bonding. Yeah, certain R groups are hydrophilic, certain R groups are hydrophobic. How do you learn this? You subscribe to SnapRevise and go through all our content, and you will be well versed in this subject and many more. Cool, great. So normally there's a little bit more that we'd go through in another lesson. We don't really have time to go through it now because I want to just talk you through the website and then also offer you the promo code for so you guys can get a discount for starting. So yeah, I hope you guys enjoyed the lesson and hope you guys learned something. Don't disappear just yet. And I will, oops, no, I need to share still. I will talk you through our package. Discard. So there are not two Ollie Vaughns. This is well spotted. This is a different Ollie Vaughan to last time. I'm just using his account. But yes, if you joined for the last one and you're wondering if you're just going crazy, you're not. It's a different person. So this is the website. It's it's been redesigned around a year ago, and it's looking very snazzy. I don't need to join a new subject. What am I doing? So I'm already subscribed, so I can go to my course. You guys, if you want to check it out, by the way, there's also a free week trial on all the packages. So I've gone to A-level biology. I'm on the AQA exam board, but we have packages for all the exam boards. So it doesn't matter if you're not AQA, this is just a demonstration. The whole spec, we've got year 12, year 13, the practicals, and some mock papers for you guys to do. We'll just go to year 12 today. I guess you might as well go to proteins because that's what we looked at. So the whole thing is organized by topic. So sort of going through the spec in the same order that the spec would go through the topics. So more or less the same order you'll have been taught these subjects in school. So let's go on proteins, which is here. Each of the individual modules, it starts with a test. So it's sort of like a diagnostic quiz just to see how much you already know about the topic. So they're not very long, like this one's 1 to 3 minutes. And you can skip them if you just want to get straight into the topic. You answer a few different questions, and I'm just clicking at random because we need to get, oh, God, that's not bad for random clicking. Yeah, you get the idea that we, sorry, I'm not looking at the quiz too much. Yeah, the diagnostic quiz will sort of give you a level as to where you're at. And I'm just going to skip the rest because there's too many questions. Oh, why did I pick such a long one? Okay, we're going to have to go through them. I should have picked one that had a shorter diagnostic quiz. Skip, skip, skip. Maybe I can just quit. Okay, perfect. Yeah, so you would obviously do all the questions because it's going to be beneficial for you guys. In the interest of speed, I'm just skipping over that. And that brings us in to the main chapter. So this is just the chapter on proteins. So there's one of these for each of the individual subjects. So there's loads of content. You've got some videos, and these go through the different content. So yeah, another one here. You've got another quiz, sort of like a midway assessment, and then some more videos on more of the content. A final quiz, and then some exam questions. So like a bank of exam questions on all on the proteins topic. So you can really test yourself. If you're on the ultimate package, on any of these videos, if there's something you don't quite understand, so you've watched the video and you're still not understanding it, you can write a comment, and someone from SnapProvise will answer your question. So if you've watched it and you're still not understanding it, and it happens, you can look at something a few times and you read the textbook and you're still not sure what's going on. It's really annoying because you can't get your head around it. You can just comment, and one of us will answer your questions for you. The colors on these videos, this bit underneath, this relates to your progress in the quizzes. So if you've done well in a quiz and you've answered all the questions on that part of the topic correctly, this will light up as green. So you kind of know already that you know that bit quite well, so you can skip it. So this is really important because the biology spec is so huge. I would argue it's the biggest of all the exam specs. Maybe some other subjects would argue differently, but it's a massive spec. So you can't be wasting time looking over things that you already know. So this diagnostic tool is vital. So if you've answered questions correctly, this will go green. You know you can skip it and move on to the bits that you don't know so well and just really target your revision to those areas. So for each thing, yeah, you've got these questions you can practice. You've got a revision guide for each topic, which look a little bit like this. It's not a very good view, but just sort of like cheat sheets, summarized all the important bits in one page, really useful. And yes, these are basically available on all the different packages. If you want to join the ultimate package, which is the ultimate quality, you also get web classes with that. So these web classes, there's 4 of them a week. Well, there's 2 web classes and 2 drop-in sessions. The web classes will be pretty much like what we did today, like going through a topic, having you guys answer questions. You can ask me questions, and then we do some past paper questions on the stuff we've just learned. That's the sort of format for a web class. So we've got those, and we've also got drop-in sessions, which I can't find where they are, but the drop-in sessions somewhere. They're basically an opportunity for you guys to ask, like, if you want to go over a specific topic. The web classes follow a sort of structure. We go through following what you're learning in schools, more or less. The drop-in sessions are, you can just ask a question on anything that you want to know about. So maybe you've been trying to revise, let's say, mitosis and meiosis at home, and you're finding it sort of tricky, or you're pretty happy with how your revision's gone, you want to test yourself. You just send a message to me or to SnapRevise, and we'll do questions on what you want to go over in those sessions. So they're more flexible sessions. They're completely dictated by you guys. So any questions on that? Sorry, I just realized I haven't been looking at your questions for about 5 minutes. I was just monologuing. £10 a month. There's a few different packages. Yeah, ultimate one is £50 a month. I'm going to go back to this PowerPoint, and there's some more information on that in here. Oh, I thought it was information about the different prices of the packages. Never mind, that's on the website. How do I end this? Website. So the packages are here. So yeah, we've got three main packages: basic, pro, and ultimate, and this is what you get for each one. So they all have those questions and diagnostic. Oh no, sorry, they don't all have the diagnostic quizzes. Only the medium package has those. They all have the videos and the mini revision guides. And the difference between the pro and the ultimate is that you get access to these web classes and drop-in sessions, and you get the option to ask questions on the videos if you're not sure what's going on. There's no buy websites for EdXLB. We have EdXLB on our website, so this is your lucky day. This is maybe the one that does cater for EdXLB. But yeah, I do feel for people doing EdXLB. It's less common than the other ones, so that most of the resources are targeted at AQA and OCR. So it is a little bit tricky for you guys doing EdXL to find stuff to go over. We also have these YouTube sessions every week. So each subject will come up around once a month. So yeah, feel free to join those. Like, subscribe to the YouTube channel, set a reminder so that you don't miss the sessions, because those will be running. And finally, the coupon code you've all been waiting for. This week, if you use this coupon code PROT10, not a very nice word. It's meant to be proteins, but it's shortened. So PROT10. It expires at midnight tonight. So if you want to do it and you want to get £10 off your first month, use it today. Use it or lose it. But you get a week trial as well. So you could combine the two. You can use the subscription. You can use this code to get £10 off, and you get a 7-day free trial. Cool. So sorry, it's disappeared. Yeah, any more questions? Those prices are for each month. Yes, Abdullah. Yeah, you get £10 off though with this code. Gunnar, unfortunately, we don't have CIE just yet. We are working on getting more of the exam boards. We don't have any of the international ones at the moment. So neither the CIE or the EdXL international A levels just yet. But hopefully in the future, that's something we'll be able to add to the website. Yeah, some of you guys are saying the prices are a little steep, but you're paying for like very good quality resources compared to other websites. The quality of what you're getting from SnapRevise is another level, and you're getting not just the physical resources, the diagnostic quizzes, the ability to answer questions. So yeah, compared to, you're sort of getting more of the service of what you get from private tuition, and compared to that, this is a fraction of the price for paying for tutors. Ikra, is it A-star assured? Any website that promises that, and I'm thinking of one in particular, is a bit of a dubious claim. Like if you put the work in and you really engage, you will improve for sure. Can I guarantee you'll get an A-star? No, I can't, and no one can. If they claim that, they're sort of lying. But yeah, if you engage with the material, you actually do the quizzes, look at the videos, join in, you will for sure improve your grade. I can guarantee that. Can't guarantee an A-star, I'm afraid. When we release the winner of the competition, I think it'll be announced on Instagram at some point. I don't know when though, sorry, but some point soon for sure. Where do you put the picture? You have to upload it to Instagram and tag at SnapRevise to be into the competition. Oh, winner announced tomorrow. There you go. Hassan, I can't confirm or deny these allegations. Cool. So if you guys have got any more questions, I'll hang around for another couple of minutes. Otherwise, thank you for joining. Thank you for giving up your Sunday to learn biology. It shows you've got real commitment to the subject, which I like to see. And hopefully see some of you guys on the website in the web classes. Cool, no worries. You're very welcome. Where do you send the picture? Just tag SnapRevise on Instagram. I'm throwing out words. I don't have Instagram myself, so I don't really know what I'm saying, if I'm saying the right things. But if you're more familiar with Instagram, I don't know, you have to take a picture of the web class, say you've been in the web class, and just tag SnapRevise, I believe is the format, but I'm sure you guys can interpret what that means. Cool, no worries. You're very welcome. Okay, all right. Bye, guys. You are welcome, Mackenzie. You're thinking of joining the drop-in sessions, so this will be whatever you want to go over. So if you wanted to do a drop-in session on any topic, you just email me, and we'll do that topic in the session. And you can say, like, can we do past paper questions on this topic, or can we just go through the content? We can do either. Yeah, it's totally up to you. What's the competition? Oh, did you join a little bit after the start? It's basically, you need to, let me go back to the first page. This will make it easier. Just flick through. This, yeah, if anyone else who joined a little bit after we started, this is the competition. We're giving away a free account. So it's, yeah, it's a great prize. You have to just, follow us on Instagram, share a pic of your class, of this class on your story, and tag @SnapRevise. And that's all you have to do. Okay. All right. Thanks for joining, guys. I will see some of you guys in the future. I hope so. Cool. Bye-bye.